Greetings, my name is Victor and I'm a recent graduate here at the University of Washington where I studied biology, evolution, and ecology. This last summer I've been involved with what I think is some pretty important research, and I wanted to share some basic aspects of it so that you can have a deeper understanding of this new parasite that's attacking our honeybees and even some of our native wild pollinators. My hope with this video is that it offers you some approach and some information about how to gather data and test your own colonies for this parasite, and that you may be able to upload some of this data to the National Data Bank in an effort to help gather a lot of information about this new parasite, and perhaps from there we'll be able to find some ways to counter it and protect our bees better. So what is this new parasite that we're talking about? Well, it's commonly called the zombie bee parasite. It is a true fly. It's a member of the Foridae family, which is well known for its parasites. Genetic confirmation has placed the species as Apocephalus borealis. It was first reported in 2012, just 4 years ago, and out of a paper published from the State University of San Francisco. Since then, its infection and parasitism has rapidly expanded across the United States, and just last week we had our first case announced in Canada. Our own research here at the University of Washington this summer showed incredibly high infection rates. Our average apiary infection rate was over 25% and spiked all the way up to 58% in the peak of the season. So if you're a beekeeper, I'm hoping this raises some red flags and alarms for you, and you feel motivated to test your own apiaries and colonies for this infection. So let's talk about how you can do that. Okay, in order to test for this parasite, you're going to have to take some samples of bees from your apiaries. There's a few approaches to doing this. I'm going to put links in the information section of this video that will help you build homemade light traps or homemade bee vacuums or things that will help you collect these bees. So, okay, 3 approaches to doing this. You can simply suit up and get into your hive and brush some bees into a jar and put it aside and let them expire, and you can do just sampling in that way. You could get a bee vacuum and you could do point sampling in your hives, so you could sample strict nurse bees or foragers or bees coming in and out of your hive, and this is a way to get a more clear picture of which bees are infected. And then the third way, which I believe is the easiest and most effective, is setting up a night light trap. Now, for some reason, we see that bees are not doing that well. They have problems with their circadian rhythm, and they come out of the hive at night, which is an unusual behavior, and they're attracted to light traps. So if you set out an entomological night light trap, you're going to attract bees that are not doing well. Not all of them will be infected with specifically this parasite, but our finding is that actually many of them are. So again, in the information section, I'm providing a link of a website where it'll show you how to build a light trap, and then I'll put another link down there of where you can buy a light trap. So you want to set your light traps out at night, about 15 to 20 feet from the hives. Let it sit out there all night, and in the morning go and collect what's in the collection jar. Go ahead and put a lid on that collection jar and let those bees finish expiring. And then our method that we found that works best so far is that you're going to take each one of these bees and put them in a gel cap. You can buy, this is like pill gel caps. You can buy these from Amazon or from suppliers all over for people who make like herbal remedy gel caps. You want to put a bee with tweezers into each one of these gel caps, put them in their own box, date it, and let them sit in an environment that stays around 25 degrees Celsius. And you're going to want to check on them intermittently for larvae going into pupation. And you're going to need something that magnifies, so a handheld lens or a scope or perhaps like a jeweler's loupe or something of this nature in order to count the larvae and the pupae that come out of the infected bees. They're simply pretty small, and it's difficult to get an accurate count without some sort of magnification. So now that you have some ideas of how to get rolling with the traps, let's talk a little bit about the biology, what's happening to your bees, what to expect while you're taking data, what are some good data points to take, and how to upload this into the National Data Bank. To begin with, what's going on with your bees and how are they getting parasitized? Well, we believe that this parasite works in what we call a wait and ambush way. That means they're likely hanging out around flowers or some vegetation, waiting for their host to come in and be busy working and distracted with collecting nectar and pollen. They swoop in, land on the bee, inject the ovipositor into the bee's back on the abdomen, lay some eggs, and take off. These eggs are thought to hatch really quickly. The larvae begin eating the insides of the bee's abdomen, and then as the larvae grow, work its way up through into the thorax, where it hits that highly nutritive flight muscle that's dense in proteins and things that larvae need to finish off their development. At that point, they exit out through the neck, as you can see here in this large hole, and they go into pupation. So this is what it looks like when the larvae come out, and you can see them moving around, and they quickly will go from that and to try to set up into their pupation. They then go into pupation, and you'll see these very sort of orange rust-colored capsules. We tend to see between 1 and 13 larvae come out of a bee. So there may be a few, there may be a lot. It depends on how many eggs and how healthily these eggs and larvae developed. Before we go on into what data you should collect in this endeavor, I want to briefly talk about getting a positive ID on the parasite from your sampling. Now, a lot of us are not entomologists, and it can be difficult to identify really small insects. So the key and the trick is to look at their wing venation pattern. Here on this slide, I'm showing you how it can be tricky and how it'll be easy to identify. So the top photo here is the parasite Foridae that's attacking the bees. The photo on the bottom is actually a fly that came out with bee samples that I brooded. So you can see how it could be confusing. But if you look at the wing venation pattern on the bottom fly, it's very, very different than the wing venation pattern on the right of this slide. So again, you're going to have to take your samples, capsule them off, put them into brood. Eventually the pupae will come out as flies, and flies will emerge. And then you want to take those flies, get a good look at their wings, and see, does it match this pattern here that's provided in this video? If yes, then you'll know that the pupae that you see in your capsules are the pupae you seek. So make sure you have a positive confirmation before you start collecting a bunch of data that's incorrect. Okay, let's go on to talking about that data. As citizen scientists, you're going to have to wrangle with data a little bit. Sure, it's helpful to say, yeah, we have a positive infection, but it's really a lot more helpful if you can take down some specific data points, put it in an Excel spreadsheet or go to Google Sheets and put it there, and then upload that to the National Zombie Watch Center. And the more homogenized and specific you are in the data, the more helpful it is to the greater cause of collecting as much information about this parasitism so that we can come to some conclusions, get some funding for research, and find some ways to help our bees. So let's talk about the data to collect. First of all, you're going to need to have the dates of collection. That's really important so that we can see the seasonality of when these infections are occurring. Then you're going to need to number your apiary replication. Now, many of us have more than one beehive and normally more than one place that we're tending our hives. So making sure that you have those separated in your data collection and that you're keeping your data succinct to those specific spots. This comes to the next point, which is getting GPS for your apiaries. Now, some of you might not have a GPS system, or maybe you don't have it on your phone, and it seems a little overwhelming to get that, but it's pretty easy. If you just go online and you type in Google GPS generator, it comes up with maps. You can put in the address and just hit generate GPS, and you can get it there. It's really easy to do. This is really critically important data. The next thing that you're going to want to take down is the number of bees, the number of total bees that was in your traps. And then you're going to move on after you work with brooding out the sample that you took. You're going to take down the number of pupae. And then after that, the total number of bees infected from your sample. And from that, you could calculate a simple percent of infected bees. And it's very helpful to also take down notes on field conditions. We have different environments regionally. There's different, ecosystems across the United States. So field conditions, where you're at, written down in notes would be really helpful. So these are basic data to collect. I recommend keeping it in a spreadsheet. It's really easy. Then you just have one file to upload to the Zombie Watch group. Okay, now that you got data down, you got your traps set, let's move on to talking about Zombie Watch in this national effort for citizen science. Okay, to wrap up here now, you've taken your samples, you've put out your traps, you've collected your data, and you need to upload it somewhere. Where you're going to do that is a website called Zombie Watch. I've put the link in the information section below this video. Zombie Watch is ran by the group that published the original research in finding this parasite, and they've set up the website, as you'll see when you go there, to be a citizen scientist stopover. You can join the group, you can upload your data, you can see tutorials on how to do your light traps, as I mentioned. You can see real-time updates of what's going on with this parasite, and you can be in contact with the researchers themselves if you have further questions. Also, if you have questions, feel free to put them in the comments section below this video. I'll do my best to answer them or to direct you to people who can answer them. I hope this helps get you concerned, aware, and excited about being part of the solution by being a citizen scientist and taking data. It's really critically important to the success of this project that there are people all over taking samples and finding out how deep this infection is running and how quickly it's spreading. So that's the end of this video. I would like to acknowledge my teammates who helped me in this research this summer, Allison and Zach, and our research mentor, Dr. Evan Sugden, who's an entomological professor here at the University of Washington, as well as the group at Zombie Watch who have been collaborating with us and helping guide us in our own endeavors, and also all of the collaborators here regionally in the Puget Sound who've allowed me to come to their apiaries and sample. Thank you, and please spread this video around for the greater good of the bees.